Welcome everyone. In today's discussion, we will be welcome everyone. In today's discussion, we will be studying the functions. We will be studying the functions and significance of phototropin, a pivotal photoreceptor in plants. Let's discuss its role in processes such as phototropism, chloroplast movement, and stomatal opening, as well as shedding lights on how plants respond to and utilize light signals for optimal growth and adaptation. Let's start with the discovery of phototropins. The early efforts to identify mutants in Arabidopsis with impaired responses to blue light, a research endeavor later expanded by Winslow Brick and his colleagues. Their work led to the isolation of non-phototropic hypocotyl and pH mutants, that is characterized by diminished responses to low-intensity blue light. The subsequent cloning of the NPH1 locus unveiled the photoreceptor responsible for phototropism, aptly named phototropin. This protein not only mediates phototropic responses but also regulates various blue light responses, collectively enhancing photosynthetic efficiency and facilitating plant growth, particularly in low light conditions. These findings emphasize the intricate regulatory roles of phototropins in plants' adaptive responses to their light environment. Phototropin serve as a blue light that has a wavelength of 400 to 500 nanometer photoreceptor in plants and is classified as flavoprotein due to its reliance on the flavin chromophore FMN, that is flavin mononucleotide. The terminal portion of phototropin encompasses two love domains, each capable of binding FMN, while the C-terminal region contains a kinase domain characterized by its serine-threonine kinase activity. Positioned at plasma membrane, phototropins functions as serine-threonine kinases. Therefore, phototropin with its flavoprotein nature and distinctive domain structure operates as a blue light photoreceptor at the plasma membrane, playing a crucial role as a serine-threonine kinase in plant responses to light stimuli. Similar overlaps in the function of the PHOT1 and PHOT2 photoreceptors are observed for their blue light responses. In Arabidopsis, including chloroplast movement, stomatal opening, leaf movement, and leaf expansions. Together with phototropism, these processes integrate efficient light capture and carbon dioxide uptake for photosynthesis. Consequently, growth of phototropin deficient mutant is severely compromised, particularly under low light intensity. Coming to the structural domain of phototropin, in contrast to cryptochrome, which are predominantly localized in the nucleus, phototropin receptors are associated with the plasma membrane, where they function as light-activated serine threonine kinases. Moreover, phototropin contains two light-sensing light oxygen voltage, that is love domain. It may be love 1 and love 2, each binding a chromophore flavin nucleotide that is called as FMN. Blue light irradiation oblique photoexcitation of protein bound FMN causes a conformational change of phototropins that triggers autophosphorylation and start the sensory transduction cascade. This figure illustrates the domain structure of Arabidopsis thaliana phototropin 1 along with three related blue light photoreceptors found in plants are algae, that is neochrome, Z-loop and aureochrome. As you can see, phototropin typically consists of two photoreceptors domain known as love domain and a serine threonine kinase domain, while neochrome is characterized by a combination of love histidine kinase domain. Moreover, Z-loop contains a love domain and a domain associated with the F-box protein, and aureochrome possesses a love domain and a basic region helix loop, that is BHLH domain. Phototropins comprises two light sensing domain, that is light oxygen voltage domains labeled love 1, love 2. Each love domain binds to a chromophore flavin mononucleotide here. In the absence of blue light, one FMN molecule is non-covalent. FMN molecule is non-covalently associated with each love domain in the dark state. Upon exposure to blue light, a photochemical reaction occurs causing a conformational change in the love domain. This change leads to the formation of a cysteine flavin covalent adduct as the FMN molecule becomes covalently linked to cysteine residue in the phototropin molecule. This covalent modification is reversible and serves as a signaling mechanism, enabling phototropin to sense and respond to blue light stimuli. The conformational shift triggered by the covalent adduct is pivotal in initiating downstream signaling events, ultimately regulating phototropic responses in plant, as you can see in the figure. From this figure, it is clear that in the dark state, the FMN chromophore in phototropin has a maximum absorption around 450 nanometer. Upon exposure to blue light, a photochemical reaction initiated, resulting in the formation of a covalent bond between FMN and a cysteine residue. This process induces a shift in the absorption maximum to 390 nanometer through a love 60 intermediate form. Importantly, this reaction is reversible in dark reaction. Here it is. This can be reversed in the dark condition. The covalent bond formed during blue light exposure allows phototropin to sense and respond to the light stimuli, while the reversibility in the absence of light enables the protein to return to its original state. As you can look, figure first displays a crystal structure of OAT1 LUB2 domain in the darkness. The protein is in yellow color and the FMN cofactor here is shown in the blue color. The Ja helix is on the left of the love domain. The three-dimensional structure of the LUB2 domain resembles a closed molecular hand holding the FMN tightly by non-covalent interactions inside its core. Moreover, this second figure illustrates the formation of the covalent bond between the flavin cofactor and a cysteine residue in response to blue light. Phototropins basically serve as key regulators in promoting phototropism, chloroplast movement, as well as stomatal opening by orchestrating responses to blue light in plants. Their ability to sense and transduce light signals allows plants to optimize their growth, photosynthetic efficiency, and water balance in varying light conditions. Now we will discuss each physiological role facilitated by phytotropin. The assessment of phototropism in Arabidopsis seedling serve as a bias to measure phototropin activity. In the wild type, both LUB1 and LUB2 domain are present. Thus, it's shown normal phototropic response. However, mutagenesis experiments were performed as shown in the figure. The LUB2 domain in particular is essential for blue light induced kinases activation and autophosphorylation of the phototropin receptor. Mutating the conserved cysteine in the LUB1 domain of FOT1 does not affect phototropic responsiveness, whereas the equivalent mutation in LUB2 abolishes the response. Therefore, it suggests that LUB2 in controlling phototropin function. This is due in part to the position of LUB2 within the phototropin molecule, where it is coupled to a protein region known as the Ja helix that is important for propagating light-driven changes within LUB2 to the kinase domain. This figure is the model for the blue light induced autophosphorylation of phototropin. It involves two love domains as depicted this yellow color in the figure and kinase domain which is depicted here as the red in color. These domains are separated by an alpha helical region or J alpha. In the dark condition, in the dark, the N-terminal region inclusive of the left domains functions as a cage suppressing the activity of the kinase domain. When blue light is absorbed by the left domains, primary photochemical changes occur, leading to the uncaging of the kinase domain and its subsequent activation. The J-alpha helix loses its helical structure as a result of photoexcitation. This activation of the C-terminal kinases domain initiates receptor autophosphorylation on multiple serine residue. Then the Ja helix undergoes a complete loss to its helical structure, and autophosphorylation within kinase domain is essential for triggering all phototropin mediated responses in Arabidopsis. In the dark, dephosphorylation taken place, resulting in the inactivation of the phototropin. Coming to the next, phototropism is the phenomena where plants exhibit directional growth in response to light. There are alterations in oxygen mobilization as shown in the diagram. Phototropins acting as crucial photoreceptor coordinate the establishment of an oxygen gradient within the plant. When exposed to light, the plants tend to bend toward the light source. This bending is facilitated by the manipulation of oxygen concentration, with the darker side of the plant exhibiting a higher concentration of oxygen compared to the illuminated side as displayed in the figure. This localized increase in the oxygen concentration on the darker side prompts rapid cell division and elongation in those cells, promoting the bending of the plant towards the light. The differential distribution of oxygen under the influence of phototropin thus plays a pivotal role in coordinating the phototropic response in plants. Phototropin also regulates the movement of the chloroplast. As you can see in these figures, it illustrates the chloroplast distribution pattern in Arabidopsis palisade cells in response to different light intensities. In condition of low light, chloroplasts tend to aggregate near both the upper and lower surface of the leaf palisade cells, effectively optimizing their absorption of light. In intense light conditions, chloroplasts relocate to the lateral walls of the cell, aligning with the incident light. This movement minimizes light absorption, prevented potential photo damage. Whereas during the dark condition, the chloroplast shift to the lower part of the cell, as looked in all the three figures. Moving forward, this figure shows that Arabidopsis FOT1 mutants have a normal avoidance response and a poor accumulation response. In contrast, FOT2 mutants lack the avoidance response but retain a fairly normal accumulation response. Cells from the FOT1/FOT2 double mutant lack both the avoidance and accumulation responses. These results indicate that FOT2 plays a key role in the avoidance response and that both FOT1 and FOT2 contribute to the accumulation response. The discovery of Arabidopsis mutants with impaired chloroplast avoidance response led to the identification of a novel F-actin binding protein called chloroplast unusual positioning 1 called CHUP1, CHUP1. This finding aligns with earlier research indicating that changes in the cytoskeleton are involved in chloroplast movement. CHUP1 is located in the chloroplast envelope and plays a role in positioning and directing chloroplast movement. As you see in figure which illustrates chloroplast movement in Arabidopsis, both FOT1 and FOT2 contribute to the accumulation response and are situated at the plasma membrane. FOT2 is responsible for the avoidance response and is also found on the chloroplast envelope. In bright sunlight, CHUP1 in cold to the plasma membrane through protein interactions binds to the chloroplast envelope. CHUP1 recruits G-actin and actin polymerizing proteins to elongate an existing F-actin filament. Later, CHUP1 and the chloroplast are pushed by the inserted G-actin, generating the motive force for the movement of the chloroplast. As shown in the figure, the guard cell pumping hydrogen ATPase play a central role in the regulation of stomatal movement. The blue light activates the hydrogen ion ATPase. Activation of the enzyme involves the phosphorylation of serine and threonine residue of the C-terminal domain of the hydrogen ion ATPase. A regulatory protein termed 14,3,3 protein has been found to bind to the phosphorylated C-terminus of the guard cell hydrogen ion ATPase, but not to the non-phosphorylated one as displayed in figure. The 14,3,3 proteins are ubiquitous regulatory proteins in eukaryotic organisms. Moreover, in plant, 14,3,3 proteins regulate transcription by binding to activators in the nucleus, and they regulate metabolic enzymes such as nitrate reductase. Only one of the four 14,3,3 isoforms found in the guard cell bind to the hydrogen ion ATPase, so the binding appears to be specific. The same 14,3,3 isoform bind to the guard cell hydrogen ATPase in response to blue light treatment. The 14,3,3 protein dissociate from the hydrogen ion ATPase upon dephosphorylation of the C-terminal domain. This figure illustrates signal transduction events following phototropin excitation at the God cell plasma membrane. The membrane associated God cell specific protein kinase called blue light signaling 1 or BLUS1 is phosphorylated by FOT1 and FOT2 redundantly. Arabidopsis mutants lacking BLUS1 show no blue light induced stomatal opening but are not impaired in other phototropin responses, including phototropism and chloroplast relocation. This phosphorylation event is essential in initiating the early transduction events that ultimately lead to phosphorylation and activation of the hydrogen ATPase. The signals from BLUS1 converge on protein phosphatase 1, an intermediate signaling molecule that activates the hydrogen ion ATPase. PP1, a serine threonine protein phosphatase, consists of catalytic subunit PP1C and a regulatory subunit PRSL1 or PP1 regulatory subunits 2 like protein 1. PRSL1 molecules modulates catalytic activity, subcellular localization and substrate specificity of PP1. PP1C positively regulates stomatal opening, broad, or you can say the stomatal opening through blue light signaling between phototropins and the plasma membrane ATPase in God cells. The figure illustrates the excitation of phototropins at the God cell plasma membrane following a series of early signal transduction events. They are firstly, phototropin absorbs blue lights leading to their autophosphorylation. Subsequently, the activated phototropin phosphorylate BLUS1. This sets off a cascade as BLUS1 signaling then regulates the PRSL1 subunit of PP1C, a protein phosphatase. Under the influence of this signaling, PP1C in turn modulates the activity of an unknown protein kinase. The activated unknown protein kinase promotes the binding of a 14-3-3 protein to the plasma membrane hydrogen ion ATPase, thereby stabilizing the proton pump in active state. This in turn results in membrane hyperpolarization, facilitating the uptake of potassium ion. And the consequence decrease in water potential instigates water uptake, culminating in the opening of stomata. These molecular events demonstrate the intricate signaling pathway that translates phototropin excitation into physiological responses, particularly the regulation of stomatal aperture. Certainly, in our discussion, we learned about the multifaceted world of phototropins and their significant role in plant physiology. Phototropins are blue light photoreceptors that act in regulating various plant responses. We have discussed their involvement in phototropism, where plants exhibit directional growth in response to light, and how phototropin regulate changes in oxygen mobilization to drive this phenomena. Additionally, we discuss the intricate connection between phototropins and other physiological processes such as chloroplast movement and stomatal opening. Phototropins influence chloroplast positioning to optimize light absorption and regulate stomatal aperture, demonstrating their broader impact on plant adaptation to varying light conditions. Thank you for engaging in this discussion of phototropins.